The purpose of this short discussion is to review material from the three papers that we're reading assignments for today. But before I do that, I'd just like to talk a little bit in general about scientific literature and reading scientific papers. Some of this you may have already realized, but how you approach a paper and how much time you spend on it, what you do as you're reading it, and how you assimilate the information is really dependent on what you want to get out of the paper. If it's something that's very closely related to work you're doing, you may need to read it and reread it and go over. You may even need to dive into the materials and methods to see if there are differences in what they're doing versus what you are doing or what you want to do. And so it can be quite a time-consuming and intense experience if you really need to get into a primary research paper. Many of the papers we're dealing with in this class are not primary research papers. They're secondary, meaning they're reviewing or giving you an overview of material work that's already been done and kind of either kind of talking about what the consensus is or giving a range of different ideas in a specific topic. So for classes, when you're approaching a paper, you kind of want to be in that middle ground. Sometimes when you're reading a paper, it's enough just to look at the abstract, right, and get a general idea, okay, this is what they're doing. If the abstract is well written, that can often be enough to give you the information that you need. Unfortunately, not all abstracts are well written. So, for instance, in an abstract, you might see something like, we did an analysis using 50 species of insects to understand relationships. That's not a very helpful statement to the reader. It gives you some general information, but you could just as easily say our analysis of 50 insects and then tell what the results were to give a summary. That's the idea of an abstract. So well-written abstracts give you the bird's eye Cliff's Notes version of a paper, and sometimes that's enough. Now, papers you're reading for this class, I would be somewhere in the middle ground. Some of them you can kind of just skim over and then maybe go back to if something is raised in one of these online lectures or you're uncertain about the topic, but you don't need to feel like you need to spend lots and lots of time on them. We don't have a textbook for this class. That's a nice thing in some ways. You don't have to spend the money. But the disadvantage is we have to get the information from kind of disparate sources, and some of them are way, way in depth, a little bit more than we need to, and others are a little bit general. So let's talk about the three papers for today just very briefly. I'm going to give you again a kind of a bird's eye view. The abstracts are pretty good. Those probably would be enough, but maybe we'll get into them just a little bit more. So the first paper deals only tangentially with our subject of models of evolution. And this is the idea of when we are doing an analysis, and they're focusing on phylogenetic analyses, which we'll spend a lot of time on, but this might apply to other analyses based on DNA data. Do you combine all of your data into one large data set, or do you do separate analyses and then somehow summarize the results of those into one single take-home message? Now, when you are using some sort of formal way to summarize disparate results. So let's say, for instance, you have a data set with 18S genes, and then you have a data set with a protein coding gene. And if you do separate analyses of those, somehow you've got to combine, summarize the results of those and tell where they're congruent, where they agree, where they disagree. If there's a formal way to do that, you could call that a meta-analysis. Meta-analyses are analyses of analyses, and so they're one more step removed from primary data, and that in and of itself can be problematic. So when we're talking about phylogenies, the data that we're primarily relying on these days is DNA data, and it's these alignments that we've begun to work with, and you've looked at in the first little project. And hopefully you've got a fairly idea of what we mean when we say data or a data set. And so the take-home message of this is there are basically two general camps for when you've got different data sets. Do you combine all of the data and make one large data set and then do your analysis, or do you do separate analyses? And then kind of as a separate point, there's a little bit at the end where they talk about, well, maybe we could use some sort of a test. They call this testing for data heterogeneity. We could use a test to determine whether or not the data sets are compatible. Maybe if we had some sort of a metric to measure if data set A is compatible with data set A, we run it through that test, and if it passes, then we combine the data, right? So we make one large data set only if they pass this homogeneity or if they are homogeneous enough to pass that test. Otherwise, then we do the separate analyses, and then you're still left with this secondary problem. So there are advantages and disadvantages to both of these. Now, one of the commonly used tests in phylogenetics and other genetic-related things is the likelihood ratio test, or abbreviated an LRT. And we'll get into it a little bit in a later discussion, but for now, just know that this uses models of evolution and then comes up with a number that we can statistically test. Basically, it's a statistical test to determine whether quantity A is similar enough to quantity B that we can assume that they are not significantly different from one another. So they're using this in the aspect of a heterogeneity test. Is data set 1 significantly different from data set 2? If so, then maybe we need to treat them differently. If not, we can combine them into a single analysis. So this, again, as said, was tangentially related to models of evolution, because we need to infer a model of evolution to do that rate heterogeneity test. Okay? And so we see inconsistent ones that are consistent with each other. Now, this inconsistency versus consistency is a little bit of a sidetrack, and we'll get into it in a later one. So don't worry too much about it. If you want to read the box number 2, gives you one example of kind of an inconsistency. And then they go through an example of when somebody did this test and then decided to combine data. Now, other people, just on a kind of a theoretical framework, say you should always combine data. That's the best approach, because you go only from your primary data, then you can do a single analysis on it, rather than multiple analyses and then trying to figure out somehow to combine that. So that's more of an epistemological or kind of a theory of how you do research approach, which is that we should our analyses should be as close to the primary data as possible. So someone may say it doesn't really matter if they're heterogeneous or not. Based on the way that science should be done, we should always combine data, even if they are inconsistent. Maybe then we just need to modify our analysis of how we would analyze, and maybe have multiple models of evolution for different data partitions. So your 18S might be able to have a different model of evolution than your protein coding genes, but yet we still all combine them into one data set to get our final analysis. Okay, so that's kind of the depth and details of what you need to know from the paper. And so the conclusion here says, although it's widely understood that data from different partitions often evolve differently, the influence of those processes that has on phylogenetic reconstruction is sometimes not appreciated. And then they go into these 3 approaches where we either always combine, we always do separate, or we do this kind of, well, let's do a test to see whether we can reject or accept the idea of enough similarity to combine data. Most analyses now combine data. There are still problems with that, more so in the past, where we may have different species represented in different analyses. And so combining data all together before we do the analysis can be really tricky if we don't have enough overlap of the species that are represented in those analyses. Okay, so there's still not 100% consensus, but I think generally people would say we do as much combining of data as we can. Maybe we do a test just to make sure, but many people would say, the test is not really relevant because we're going to combine no matter what. And as our data sets get larger and larger, we're having more complete sampling of different species. Combining data is more, it's easier to do because we have the same species represented for many different genes, and we don't need to do separate analyses because we have different species representation. Okay, so that's paper 4. Paper 5, again, is a little bit of an aside, and doesn't deal directly with models of evolution, but indirectly it's kind of important because it talks about common genes that have been used historically for DNA analyses. Now, this is now about 6 years old, and so it's a little bit dated. And as data generation continues to speed up and we're having more and more and more sequences, we're really taking more of a full genome approach. And so before, it used to be that you would go out for you begin a study, maybe get some grant money, and you would say, I'm going to target a certain number of genes for a good sampling of species across my group of interest. And originally it was 1 or 2 genes. That was 20, 30 years ago. And then, a decade ago, it was more like, okay, let's do 5 or 6 genes, and then say, oh, I can do 20 genes and target those 20 genes. We've now really kind of switched over where many of the analyses now say, well, let's sequence the entire genome, or at least a transcriptome. If those two, the differences between those, if you're in your head you're still, I'm not sure, let me give you the 1-minute version. A genome, of course, is an attempt to determine the sequence of every single bit of the haploid genome. So for humans, that's 3.2 billion base pairs, give or take, and you'd have the entire genome. You would maybe then have to identify genes with the human genome. That's fairly well established already. A transcriptome means you're only going to be sequencing genes that are expressed in whatever tissues you're sampled. So you only get genes out of it. So instead of the whole 3.2 billion base pair genome, you get only the 1.5% of the genome, and probably not even all of that, because not all genes are expressed in every tissue. So you'd only get the genes, but the genes are usually the most conserved and the most easy to work with when we're doing some sort of comparison between species. So the approach now is to really kind of sequence, because it's cheap and fast, much more so than in the past, sequence as much as you can, and then use all of that data and pull out the genes that you feel might be useful, either because they've been established in the past, which this paper talks about some typical genes that are used for different groups of organisms, or because you've done some sort of analyses and represented them. Now, one of the big issues of this big data approach is trying to separate out genes that are orthologous versus paralogous. Okay, and so basically one of the main criteria is that if we use paralogous copies of genes to try and infer relationships, we're going to be tracing the gene history rather than the species history. Now, if that doesn't make sense to you, go back. You can review our little discussion about orthology and paralogy and where they trace their common ancestry. But if not, just kind of put a pin in that, and as long as you know that we should really use orthologous genes for understanding species relationships. And we'll dive into that a little bit deeper in a later chapter. But for now, realize that could be a big problem. So for instance, if you're lining up alpha-1 hemoglobin with a beta strand, like an alpha hemoglobin with a beta hemoglobin, they're similar enough, you might be able to line them up, but that's not going to be the correct data set for you to figure out species relationships. So oftentimes what people will do is they'll either use genes from gene families where the copy's orthology has been very, very well established, or they'll say, well, because we're uncertain about all the orthology paralogy issues within genes that come from a big gene family, let's pull out only the single copy genes, the ones that aren't from a gene family, and then we don't even need to worry about this orthology paralogy problem. Okay, so again, take-home message, just realize now that data is becoming a little bit different rather than a group of target genes, although these are still very important in much of our early work, which is being reinforced and corroborated by the bigger data sets in many cases. But a lot of these genes are now only a small portion of the overall genetic material that's used to do phylogenetic analyses. And then they also point out that it's important that we don't just throw away the non-DNA data. Now, the focus of this class, because it's a cellular and molecular evolution, is the DNA data. But just be aware that there still is lots of other data that you could also use for primary analysis. Okay, and again, if you're reading this paper, you maybe kind of skim over it, go through stuff. If you see math and that's not your deal, which is probably for most of you, unless you're bioinformatics, and you say, oh, 28S, maybe scan over these things. These are genes that have been and are used commonly. If you're interested in plants, you can skim this, but don't feel like you need to memorize this. You've got the paper to go back and refer to if you ever need to. Now, it does talk about phylogenetic tree reconstruction methods, so we're getting now a little bit into what we're going to jump into next week. So this part, we've already talked about multiple sequence alignment. Just be aware that the models of evolution is the topic for this one. that this is a little bit of kind of material that we're going to be getting. Some of it we've already got, some we're going to be getting later. And again, that's kind of one of the disadvantages of not having a textbook, is the order in which things are done. So if you're having elements of this that are familiar and other elements that are like, oh, that's really kind of, I'm not quite sure what they're talking about, like bootstrapping and jackknifing, that's fine. And you can use the lectures as a guide to help you know what parts of this you really need to take, what parts we might be doing later, or maybe they're just kind of beyond what we need to do for this class. Okay, so read the discussion, wrap that up. That will give you a nice, good overview. And this is, again, kind of wide-ranging, but is a nice summary of many of the material concepts that we're going to be talking about. The last one I'm not going to spend a ton of time on because this is the main topic for the first two lectures. Kind of try to give you a background in lecture 1, and then in lecture 2, we talked about many of the things that they get into here. They get a little bit more detailed with some of the math behind it and the models that are used to help us estimate whether or not we should use a model of evolution. So the only thing that I want to kind of emphasize is how you would select a model of evolution. You should review and know those 5 models of evolution, which we kind of presented in increasing complexity. You should understand the argument that we really want to use a complex model if it best fits the data, because then we can account for all the different processes that have occurred in those DNA sequences. But by choosing those more complicated models, we also have a limited amount of we have some limitations that are imposed on us because we have to estimate more parameters, which is decreases our precision in any sort of statistical analysis we're doing. So just review that. We've got some nice review of those 5 basic models here. And then the last thing that I'm going to say, well, actually, 2 more things. Models for protein are more complex and rely primarily on going back over kind of post hoc, right, is after the fact, and looking at data sets that we've got some idea about and figuring out how they've evolved. And so they're more complex because there are 20 different amino acids, but in reality, they're really, of necessity, linked to the nucleotide analyses. In nucleotides, we only have 4 DNA base pairs. We got lots and lots of data. We can kind of assess what's going on within our data set, maybe more generally what's going on in big data sets to come up with parameters for like transitions versus transversions, or to estimate what the actual frequency of a certain base pair might be. Okay, so just for now, know that amino acid models are much more complex. There are 20 different amino acids, and proteins have a much more complex way that they interact with their environment than DNA. DNA basically has one job. Protein has, depending on the protein, has many, many different possible jobs. And then they look at the end at some of the applications of this. We'll talk about some of these applications later on. If you want to just skim over this, that's fine. And then they kind of get into this idea of a whole genome-based phylogeny also, which if you're using a model of evolution and maybe you need to be able to make sure that you can partition data and say, even though I'm going to do a single analysis, I might need slightly different models of evolution for different genes because they have been under different evolutionary pressures. And so we'd like a model. We can't just come up with a one-size-fits-all model of evolution for all of the data. Okay? We'll start talking about parsimony, maximum likelihood, and some of the methods, Bayesian maximum likelihood. There are others, but the ones that implement these models of evolution. And we'll talk a little bit of how they do it. We're not going to get into the guts and the math of it, but there's some nice examples of how these models of evolution might be used both in phylogenetics and in closely related fields. So don't spend a ton of time on that, but you can review it, go over it. And then, of course, the conclusion, kind of for this mid-range amount of time we're shooting for spending on these papers, conclusions are fairly an important one. So if you've only got a little bit of time for a paper or a medium amount of time, if you only have a little bit of time, just read the abstract. If you've got a middle of time, read the abstract, go over the introduction, maybe then jump ahead and read over the conclusions. And if there's something that you're interested in or that you need to know more in depth, you could go back and spend some more time reading through the middle parts of the paper, the guts. We aren't doing a ton of primary research papers, so we don't often have materials and methods because these review papers are just overviews and not the primary literature itself. All right, if you have any questions, I'll be available on Wednesday and Friday at 3 p.m., or you can call me and make an appointment.